Hello, and welcome to the pre-lab lecture for 2130's SKITTLES lab. In this lab, we're going to learn about chromatography. Chromatography is a way used to separate different materials by the fact that they can be absorbed to a solid and or dissolved in a liquid, and the ratio between the time it's absorbed and dissolved tells us how far it's going to move in our system. So let's talk about some basics of chromatography. All chromatography methods operate on the principle that you have a mixture that will distribute unequally between two immiscible phases. So by miscible phases, I mean something like a liquid and a solid or a solid and a gas. Okay. Now in this, we can allow the thing that we're trying to separate to move in the mobile phase, but it is slowly moving past the stationary phase. And that's typically the solid or a liquid. Now, the individual components of these have affinities to the solvent, but it also has affinities to the stationary phase. For example, a dipole-dipole interaction or a hydrogen bonding interaction. Okay, so now because the stationary phase and the mobile phase have probably different polarities and different hydrogen bond abilities, you will see the same compound go into the solvent phase and then be absorbed to the surface and then be reabsorbed into the solid phase and into the surface and continuously move like that. So that ratio of time spent on the solid phase versus the liquid phase tells you the difference in which these things are going to move. Okay, and since each component has two different phases, has a different equilibrium constant or partition coefficient between those two things. And these are called migratory bands. So imagine one component is spending 20% of its time in liquid and only 80% of its time on the solid, it moves very slowly. Where if one component spends 80% of its time in the liquid and only 20% of its time on the solid, it moves very quickly. Okay, so what this looks like is that you'll have your mixture of materials here, and because your two different components can move at different rates, they shouldn't be moving at the same rate, you will see them as your liquid phase slowly moves up your solid phase, you're going to get separation between the two. And so this is the general concept between liquid chromatography. We can also do it with high pressure liquid chromatography, thin layer chromatography. We can even use it in a gas liquid phase chromatography, or even gas chromatography where you have a gas moving along a solid. So all of these different chromatography methods rely on this ratio of time spent on the solid phase and time spent in the mobile phase. Okay. So in thin layer chromatography, we typically have some kind of solid absorbed to a surface like glass or metal or plastic, and therefore you can actually have just your material be absorbed up the material that's the stationary phase, and the liquid phase will slowly capillary action up it until it gets to the end of the phase. As it's doing that, we have to measure the difference from where we started our spot and to the end of our solvent phase. The reason we do this is that we can take the ratio between where we started the sample, where it ended the solvent front, and where your components ended up. And by comparing those two, we can get what we call the RF. Okay. Now, the other thing we need to do is that in our case, we are going to be using, instead of thin layer chromatography, we're going to be using something called paper chromatography. In this case, paper is our solid phase, and we can use water as our mobile phase. And so in this case right here, we have a paper which has a lot of different groups on it to make it very hydrogen bond very well. And we are using water, which also hydrogen bonds very well. So our individual components probably are going to hydrogen bond to the paper and to the solvent, but in a different partition coefficient, so they should move up the paper at different rates. Okay, let's talk about how we're going to identify these. We're going to identify these things by what we're calling retention factors, or RF values. Okay, so when we're measuring our RF values, we have to measure the total distance traveled by the solvent, and then the total distance traveled by that particular component. And if we dissolve the we divide the distance the component traveled by the distance the solvent front traveled, we now have our retention factor, or our RF. And they should be different for many, many different compounds. Okay. So the reason we want to do this is that we're going to be able to see a very different moving from each of these different factors. So in our experiment, what we're going to end up doing is we're going to end up spotting our red, orange, yellow, green, blue, and purple dyes onto a piece of chromatography paper. We're going to develop that by putting it into a beaker full of water and watch the water slowly rise up. Now, at the end of the time, when it's about a centimeter from the top, we're going to draw a line, and that's as far as the solvent front has moved. Once we dry it, we can now take the distance traveled by the solvent and the distance traveled by the spot, and we'll able to divide the spot by the solvent, and we'll get our RF factor. Now, that's what we're going to do for all of our known dyes. After that, we're going to take our Skittles, crush them up, and extract that dye from those samples. And when we do that, we'll be able to spot the orange Skittle, the red Skittle, the yellow Skittle, and the green Skittle on that same chromatography paper. And we'll be able to process that by using that same solvent of water to get our separation. And then we're going to measure our RFs on those. Okay. So on our sheet that we're going to, on our data sheet, we'll see we have a big table. And on our big table, we have the bands. We want to do the bands from the top to the bottom. So the one that travels the furthest, you want to have that up, and that should be your R value closest to 1. And then the one at the bottom is the one that is going to be the least. And so what we're looking for is in that first column, you want to say number the top to bottom 1, 2, 3, 4, 5. 6. And so red might be a 3, yellow might be a 1, green might be a 2. So you just write down which in the order they are. And then you also calculate the RF factor, again, dividing the distance by the spot by the distance of the solvent, and that should give you your RF factor. Your RF value should always be between 0 and 1. Okay. So you're going to do that for all of your known samples first, and then after you've done that, we're going to go ahead and run the Skittles, and we're going to see what those RF factors are. And you're going to fill that out on your data sheet as well. After that, you'll of course have to draw some conclusions in your post-lab questions and make sure you upload those to Canvas. Let me show you a picture real quick of what you should see in the video. Okay, so what we'll see in the video is something that looks like this. And so you have your scale on one side in centimeters and in inches, and then we have our solvent front. Our solvent front is right here at the top of this paper. And so what you want to do is you want to take the distance traveled by the solvent front, and then the center of each of these spots. Notice this is the red, the orange, the yellow, the green, the blue, and the purple. And so this is where the center of those are. And this is where you'll calculate out on that first set of bands. Then you'll be given a second one of these where you can pause the video and calculate your RF factors for the different Skittles parts as well. Okay, go ahead and go take the quiz first, and then go ahead and watch the video on YouTube and fill in your data sheet and your post data sheet and upload those to Canvas. Thank you.